All right, so it is my delight to go to the next section. We'll have the session 1 on fundamentals, and the moderator is the incomparable Dr. Abbott F. Clark, Abe Clark, who is in the Graduate School of Biomedical Sciences, Executive Director and Farvo Professor, North Texas Eye Research Institute, the Executive Director and Regents Professor, Pharmacology and Neuroscience at the University of North Texas Health Science Center, and a remarkable, wonderful person. And I ask all of the speakers to come up here.
Well, good morning. We have a really exciting program for you where we're going to talk about the fundamentals of animal models, the advantages and disadvantages. And we have 4 terrific contributors in this area. So you're going to go first? Okay. So Michael, Michael Elliott is at the University of Oklahoma.
That's true. Oh my gosh. We don't need it.
Oh, I didn't take mine.
There you go.
Thanks for the introduction, and thanks for the invitation to be here. This is actually my first fast track, so I'm excited to learn with everyone here. I've been to the meeting in the past. In fact, I think it was at the first one, and there wasn't a fast track then, and so this is really would have been a great experience for me, and I'm excited to participate and learn. So I don't have anything to disclose, and what I'm covering and what the panel's covering are really attributes that we need for key attributes we need for glaucoma modeling in animal models. And I will point you to this excellent review from our distinguished, incomparable moderator, Abe Clark, who really covers, I think, a lot in this review of what probably the whole panel is going to discuss in some way today. What I'm tasked with is focusing specifically on the intraocular pressure pathway, the trabecular meshwork, Schlemm's canal, aqueous humor drainage pathway. And I'll be focusing on inducible models and modeling elevated intraocular pressure and pressure dependence, again with specific relevance to the outflow pathway tissue. So the outflow pathway is this as we've seen already very nicely, this sort of circumferential vascular system that has this unique vessel called Schlemm's canal, which is connected to outlet or collector channels that drain into the episcleral venous system, drain aqueous fluid. And in front of that, on the anterior chamber side, is this tissue, the trabecular meshwork, which is this lamellated tissue that has multiple layers of trabecular meshwork cells. And so aqueous fluid has to pass through all of this and then pass across the wall, the inner wall of Schlemm's canal to get into the lumen where it can drain into the venous system. And so outflow pathway dysfunction is associated, as we've seen, because of the IOP association with glaucoma. And I've sort of crudely indicated it as issues with the drain. And so these are just a few of that heterogeneous class of diseases in which we have the association with the drain. So congenital glaucomas, the drain is not developed or doesn't develop properly. Angle closure, the drain is not accessible. Pseudoexfoliation, pigmentary glaucomas, and others, the drain may be clogged with debris. In primary open-angle glaucoma, as we just heard about, the drain is open, but the drain itself may be dysfunctional. And we have to find ways to model this. And even normal tension glaucoma, there's some evidence that there's therapeutic benefit to lowering IOP even in patients with normal intraocular pressure. So maybe improving drain function is useful. And this just describes over here the relationship between intraocular pressure and the drainage with this linear increase, the trabecular conventional outflow pathway being the pressure-dependent component of the outflow pathway. So how does the anatomy of, in this case we'll talk about mice, because that's what, mostly what I'll be talking about in this talk. How does the anatomy of the mouse outflow pathway compare to humans? And I want to thank Colleen for sharing some of her slides with me for this. So the human trabecular meshwork, so what you see here are human and mouse histological sections as well as some immunostaining of the same tissue. And one of the things you can appreciate is that the human trabecular meshwork tissue is a little bit more developed. It's thicker, many more layers than what we see in the mouse. The mouse trabecular meshwork is quite a bit thinner, but it does have a lamellated meshwork. There are layers as well as a continuous Schlemm's canal, which is an advantage of the mouse model over some others. So similarities between the mouse outflow pathway and the human are the continuous Schlemm's canal, the lamellated TM, composition, for example, cytoskeletal components, the network, the connections between the trabecular meshwork and the ciliary muscle, the innervation, all of these are similarities between the mouse and the human that suggest that the mouse might be a good model. So what about function? As was already talked about, the Goldman equation, this is kind of a modified version of it, but this Goldman equation basically describes the relationship between intraocular pressure and the components that lead to that, including the aqueous formation rate, or FIN, the uveoscleral outflow, which is not dependent on pressure, and then the C, which is the outflow facility, so that's a description of the pressure-dependent component of outflow, and then the episcleral venous pressure, which again is the backfilled pressure for the system. So the important thing here is this trabecular outflow pathway is pressure dependent, and as you can see in a study from Sherwood and Daryl Overby, that the mouse outflow tissue, this is in an enucleated perfused eye, is also displays pressure dependent outflow. And in fact, in this earlier paper from Ross Ethier, Dan Stamer, and Daryl, they stated that mouse eyes are similar to human eyes and that we have no detectable washout rate and a linear pressure flow relationship over a broad range of intraocular pressures. Because of the absence of washout and the apparent presence of a true Schlemm's canal, the mouse is a useful model for studying the physiology of the inner wall of Schlemm's canal and conventional outflow tissues. Okay, so what describes, or what would be a good description of models? And so a group of us got together a few years ago and put together essentially a consensus statement that is referred to here and published on models for mouse models for ocular hypertension. And the minimum acceptable standards for this description is stated here. Mouse models used to study outflow physiology must demonstrate elevated IOP, or at the very least decreased outflow facility and open iridocorneal angles, and must include histologic descriptions of the morphology of the conventional outflow tract. So the question is, I'm going to introduce a couple of example models now that I think fit that bill. And the first one I'll start with is steroid-induced ocular hypertension. So as I think many of you know, corticosteroids are commonly used for treatments of a number of inflammatory eye diseases. But the downside, a major adverse event that's associated with steroid treatment is ocular hypertension. So having models to model that are useful. So mouse models have been developed, and these are just some lists. And so the references in my slides are actually, as you've noticed, are interspersed within the talk. So the talk's available, so you'll be able to look up some of these papers. But because of time, I won't go into all the different models. But we have systemic models of steroids, so dexamethasone treated, in this case, with an osmotic mini pump. There's been studies with topical administration of dexamethasone, as well as injections, periocular injections of dexamethasone. The study I'm going to focus on today uses periocular subconjunctival injection of dexamethasone-loaded nanoparticles, which allow for slow and sustained release. And this was studied from Stamer and colleagues. And in terms of meeting the criteria, I think we'll be able to see that this model meets it. So when dexamethasone-loaded nanoparticles are implanted, they result in an intraocular pressure, a sustained intraocular pressure elevation, as well as a reduction in outflow facility. In addition, they provide morphological description of the open angle, as well as additional measurements, including evidence of fibrosis in dexamethasone-treated angle tissues, as well as ultrastructural studies showing extracellular matrix deposition. They also did an interesting experiment where they used OCT to measure the openness of the Schlemm's canal under different clamped pressures, and showed that the dexamethasone-treated eyes are resistant to Schlemm's canal collapse, suggesting increased stiffness of the tissue. So I think we can see that this model meets and exceeds the acceptable models. So now I'm going to transition for the last part of this on viral transduction. So there's also been a number of studies to induce ocular hypertension or potentially to provide therapy using viral delivery, viral-mediated gene transfer. And so the choice, as in any virus experiment, involves considerations of tissue tropism, the size of your payload, what promoter you might use, the inflammagenicity of the viral construct that you're using, and also the site of delivery. And I think the earliest studies in the eye used adenovirus, because adenovirus has great tropism for the TM. It's efficient at delivering transgenes. And what I'll talk about are studies that deliver TGF-beta 2. But the downside of AD5 is that it has been reported to be a little more inflammogenic. So here's a study from Wei-Ming Mao's lab using a lentiviral TGF-beta 2 approach to sort of circumvent some of the inflammatory issues with AD5. And what they demonstrate in this study is an IOP increase when they deliver lentiviral TGF-beta. They show the localization of transduction, as well as showing that the angle tissue is open. So this looks like at least a promising model to deliver transgenes. The last thing I'll shift gears on is a study that uses AAV2. This is a more recent study. And in this case, they're not delivering TGF-beta 2, but are delivering means to manipulate the Tie2 system in Schlemm's canal. So they use an AAV2 that can target the Schlemm's canal, sort of bypass the TM, and also not target other vascular systems. And in this study, they used both an AAV2 that delivers a Cre recombinase, which they used in the context of a mouse that has the Tie2 gene floxed or has intragenic LOXP sites inserted, and also an AAV2 that delivers gene editing components, which I won't talk about. What they did in the study is it's a fairly complicated slide, but what they did is they delivered the AAV2 Cre intercamerally, in this case in a reporter mouse, in which when the LOXP sites are recombined, the mouse will express TD tomato in recombined tissues, and they show very nicely that they get transduction of the Schlemm's canal without transduction of distal vasculature. When they inject that same virus in the Tie2 floxed mice, the mice in which the recombination results in tissue-specific deletion of Tie2, they see a sustained elevation in intraocular pressure, as well as a reduction in outflow facility. So with that, based on the similar outflow pathway structure and function, mice are useful in vivo experimental models to study IOP outflow physiology and pathophysiology. Induction of pathology, in the case I presented corticosteroids or virus-induced gene manipulation, in the aqueous humor drainage pathway leads to glaucoma-relevant phenotypes, including IOP elevation and outflow facility reduction. Viral-mediated transduction can relatively specifically target outflow tissue, providing a strategy to gain mechanistic insight into IOP modulation in vivo. We should take care in ensuring proper control, as in all experiments, to be sure that the effects are not due to the virus and are in fact due to the genetic manipulation the virus is carrying. And finally, in addition to generating models with the viral approach, it also offers promise for therapeutic potential. And with that, I'll conclude.
We have time for questions at the end? Okay. So my pleasure to introduce Rob Nichols, University of Wisconsin.
Thanks to the organizers for letting me give this talk, and thanks to you for coming. This is a great part of our meeting. We love the participation. So I'm going to talk about optic nerve crush. I have no disclosures, and this is my first slide. And after seeing Bree McWhorter's talk yesterday, I've scrapped it, and now this is my first slide. So I want to give sort of a history of why we developed optic nerve crush in mice. And it started back in the early 90s, really, when we were pretty convinced that ganglion cells were dying by apoptosis and glaucoma. And from that, we wanted to understand what the genes were that were involved in this process, being an intrinsic program within the ganglion cells. In order to understand the genes, we needed an animal model where we could manipulate the genes, and by far the most prevalent and best model available at that time was in mice in the induced mutant resources. So we needed a way to study ganglion cell death in mice. And it wasn't just mice with glaucoma, which didn't exist at that time, but we also needed a model where we had synchrony, where we get a lot of ganglion cells activating the cell death program at the same time. So at the time, there were a lot of models of acute optic nerve damage. They were in rats or cats, or we even did it in non-human primates. Glaucoma models in mice didn't exist. We didn't realize that Simon John was actively working on measuring ocular hypertension in different inbred lines. And there was a mouse model of acute optic nerve damage, but it was really quite severe. You had to actually go through an intracranial approach. You had to suck out part of the brain to access the optic tract, and then attack the tract at through that hole in the mouse brain. And the mice did not do well in that surgery. So I had the opportunity to have lunch with the famous Albert Aguillon. He's famous. A lot of you probably don't know who that is, but he's quite famous for doing transections and exotomies and crushes on rat optic nerves. At the time, he was basically the godfather of rodent optic nerve biology. He's most famous for his work where they put in a peripheral nerve graft after exotomizing the optic nerve and having regeneration grow through that graft. And so I had lunch with him, and I said, Albert, I need to do this on mice. Have you tried it? And he said, yes, we have, and you can't do it. So it's impossible to do. They bleed profusely. Don't even bother. So I went back to the laboratory, and I talked to my postdoc, Yan Li, who is a pretty accomplished ophthalmologist who had come from China and also a very good microsurgeon. She said, well, I don't believe him. And so she started to work out the protocol. And in about a month, we had a working protocol where we could use an intra-orbital approach to crush the optic nerve of a mouse. So this is a graph and a picture from the very first publication of this. It came out in '99. What we did was we counted cells in the ganglion cell layer using sections of DAPI-stained retinas. And what we found was that we could get a pretty reproducible change in number of cells in the ganglion cell layer. It turns out we lose about 50% of the cells in that layer by 3 weeks. And we've done studies since then to show that that 50% represents the ganglion cell population in the ganglion cell layer. The other 50% of the neurons are what we'd consider displaced amacrines. Now, that was then. The more modern way of looking at ganglion cell loss after optic nerve crush is to stain the retinas with antibodies against ganglion cell specific markers. This is an example of RBPMS. This is actually 90 days after optic nerve crush. You can see in the eye that was crushed, there's very few cells remaining. These are probably the intrinsically photosensitive retinal ganglion cells. They actually disappear and then come back, so to speak. We can talk about that in a minute. But there are some caveats on how you use this model and how you evaluate the pathological effects. So I have some caution flags in this talk, and the first one is that retinal ganglion cells downregulate normal cell-specific markers prior to loss. So what do I mean by that? Again, this is studies where we had counted the loss of cells in the ganglion cell layer using sort of DAPI stained images. We thought, well, maybe we could use a marker of ganglion cells to quantify ganglion cell loss. And we started doing experiments with the best and known marker at the time, which was Thi1. And what we found in our experiments, and we're doing in situ hybridization, which is shown here. We were also doing an old-fashioned method you probably never heard of called RNase protection assays to quantify mRNA messages. What we found was that by in situ, the quantification of Thi1 mRNA showed a down regulation or a disappearance well before we saw a loss of cells. And I have here. So these two panels on the diagram here show the contralateral retina of a Bax knockout mouse. And this is important because Bax knockout mice never lose a ganglion cell. You can crush the optic nerve and go almost 2 years out, and they still have the same complement of cells in their ganglion cell layer. But even in these mice that never lose a ganglion cell, you lose Thi1 message. And so this became an important aspect in how do you quantify ganglion cell loss. And using a ganglion cell specific marker may actually be inaccurate. Had we done that in our studies with Bax knockout mice, we would have concluded that ganglion cells die in Bax knockout mice. So this graph just shows sort of a summary of data from the literature looking at a variety of different markers for retinal ganglion cells. It exhibits an exponential decay in mRNA, and in some cases proteins are shown here. The first cell loss that we typically see in the optic nerve crush model occurs somewhere around 6 to 7 days. And then I've got another marker in here called activated caspase 3, which I'm going to try and convince you is a really good marker to use. But the take-home point here is if you're going to quantify ganglion cell loss, and you're going to do it by staining for a protein, you really have to understand what the half-time of that protein is, because it's not being made anymore. You're staining latent protein, and if it doesn't persist, you're going to have a cell that's still there and may still be alive, but doesn't have that marker, and you're going to miss it. So here's a couple of alternatives. One of them is staining with an antibody called TUJ1. And in our field, we generally believe that beta-3 tubulin, which is what TUJ1 stains, is highly expressed in retinal ganglion cells. It's not exclusive to those cells, but it's a really good marker for retinal ganglion cells, and it's probably the most stable marker that we know of. So it persists for a long time. And then in this, you can see the red staining. This is actually activated caspase 3. And so that's a really nice marker that we use to show, to detect cells that are undergoing pathology. And then after realizing that markers were not so great, a lot of us sort of just switched to doing total cell counts. This is showing Nissl staining, for example, contralateral and crush eye. You can get the same kind of images on a whole mount using DAPI, and that's really fast and quick. Okay, caution flag number 2, genetics matters. So here's some data, and on this graph here, this is an experiment that we did using optic nerve crush on 15 different inbred lines of mice. And what we found was that there were mice that were more susceptible and mice that were less susceptible to the crush protocol. In fact, it was so predictable that in this experiment, we used 10 mice in each group. And here, we use about 30 mice in each group. So here's the resistant strain, showing its histogram of cell loss. Here's the susceptible strain, and then we can move the phenotype, or the quantitative trait, based on crossing these mice. So here's a cross between those two mice. We actually shift the phenotype to more the resistant strain. It turns out that this is a single locus that is dominant. If we do a back cross, we shift it back towards the susceptible, and so on. So it's a technique and a quantitative trait that is good enough to do genetics on. And this just shows when we did the genetics of a mapping population, we got a single locus, a very strong LOD score associated with it. And it turns out that it's this little tiny area on chromosome 5s, but we're down to about 1 million bases. And we think the gene is this thing called SPINK2, which acts as an inhibitor of the proteasome. Caution flag number 3. And I'm not going to go into this much, but the contralateral retina is not a control. My lab, I have been guilty of using the contralateral eye as a control for many years, and now I'm a big believer that it's not a control. In fact, that talk I'm going to give in a couple of days goes precisely to this effect. So I'm not going to talk about it too much here, but a couple of bullet points is that we know now that both acute damage and experimental glaucoma induces reactive glial responses in the contralateral retina. And there are conflicting reports that the glial responses actually affect ganglion cell numbers. So some reports, especially long-term, suggest that there's about a 15% dropout of cells in the contralateral retina. That's been refuted by a more recent paper. So it's kind of up in the air. I think the best control you can use is a naive retina in these experiments. Okay, I have no summary or anything like that, but what I want to do is show you the procedure. And so here's a mouse eye, and this is Yan doing the procedure. You put a numbing eye drop on the eye. The mouse is anesthetized already. Then she's going to go in, she's going to do a lateral canthotomy. So the nose of the mouse is somewhere in here. So she's going to crush the skin so it doesn't bleed. That's about 10 seconds that you do that. And then she's going to cut that to expose the back of the eye. Okay, so there's an important thing here. She's going to grab the conjunctiva and take some iridectomy scissors and cut the conjunctiva in sort of about a quarter half moon circle around the eye. But in this, you'll be able to see exposed the vascular plexus right in here. And this is what Albert was referring to, is if you nick that, this eye is going to bleed profusely. Okay, so she used a pair of scissors, but an important element of this is to actually scrape the surface of the sclera, because those plexi are covered by a very fine membrane that wraps up the plexi, and you really have to break open that membrane to get in there. Okay, now she's exposed the nerve, and she's used self-closing forceps, and she's clamped it. And we clamp for 3 to 5 seconds. And it really, you need to, every time you buy a pair of these self-closing forceps, you need to standardize or you have to figure out what the best clamping time is. Okay, so that's the end of my talk. And the rest of the presentation has references that might be interesting.
Our next speaker is Becky Sappington from Wake Forest University.
Thank you very much. Thank you all for coming today. I have one disclosure, which is I'm on the Scientific Advisory Board for Just No Therapeutics. Okay, so I've been tasked with talking to you about inducible hypertensive models. And this is specific to the posterior segment. And it's different than the models that Michael Elliott talked about. And I think that's probably the first key point here in the talk, is that there's a bit of a disconnect between models for studying the aqueous dynamics and the regulation of IOP, and those that are looking at examining posterior segment degeneration. And what you will see as we go through the talk is that one of the key features is actually that these models for posterior segment actually damage, for the most part, the anterior segment, which would preclude evaluation of normative function for the anterior segment. So in these cases, ocular hypertension in our models are looking at impeding aqueous outflow. And the model requirements at a very kind of simplistic and fundamental level is that in a pressure-induced model, you need to have a reliable elevation of IOP, right? And then you need to have retinal ganglion cell degeneration that can be tied to that elevation in IOP that you created. And I added to this third one, which we can discuss during the panel if you'd like later on, that optic nerve head remodeling, in addition to ganglion cell degeneration, I think is actually a feature that is shared and necessary, in that it covers things like glial cell reactivity and changes in extracellular matrix. So there are many different models, and despite the fact that we have these kind of 2 to 3 rather straightforward criteria for what constitutes ocular hypertension model for the study of posterior segment disease. And I've decided, for the purpose of this talk, to split them up based on induction method. And so we're going to talk about 4 today, 4 categories: injectables, surgical approaches, intermittent elevation approaches, and then something I'm calling other. But why do we have so many different models? Well, it's related to the variable features, right? So the things that come along with inducing this elevated pressure. And these factors are things like rate of IOP elevation, duration of IOP elevation, the magnitude of IOP elevation, and then also the secondary effects, right? So I already said that the elevation and IOP is caused often by damaging aspects of the anterior segment, and so that has a secondary impact that is arising from the induction itself, and models vary on that. There's also considerations to be had around the ability to translate a model from mice, which we've talked about quite a bit this morning, into some larger animals that you'll hear about later this afternoon. And then one of the other features that I think is important to consider, so we're going to talk about those, are skill level and equipment requirements. So for each of these models, we're going to go through, or each of these categories, I'm going to go through and hit the highlights of models that you'll see quite a bit in the literature, and where they kind of compare on all of these points. And I want to stress, kind of the second major point to the talk, that I don't like talking about pros, cons for models. It's more considerations, because pros and cons are relative to what you're looking at and the situation in your laboratory. And so I will refer to all of these as considerations. Okay, so we're going to start off with inducible hypertension models in the surgical space. So the first one is laser photocoagulation of the trabecular meshwork, the limbal plexus, or the episcleral veins. And the purpose of this laser photocoagulation is to increase resistance to outflow pathways through scarring of trabecular meshwork and Schlemm's canal. So IOP elevations, you can, from the literature, generally expect in this model is up to 90%, and that rate of increase to stable elevation can range from hours to weeks depending on your approach. Retinal ganglion cell loss, again, depending on the approach, how many vessels and what combination you choose to photocoagulate, you can have anywhere from 6% to 70% loss over a 12-week period. So considerations, this requires a laser. Diode is the most common, but the use of the laser allows you to configure your laser settings in terms of strength, as well as how many targets or what targets you're choosing to laser. And so there's quite a bit of flexibility there. There's a moderate incidence of secondary pathology, but there is a high skill level required, and the reproducibility is high once you acquire those skills. The literature also demonstrates that there's quite a bit of species and strain variabilities, and so that's something you need to consider when you're looking at how you would want to apply this model. The second is episcleral vein cauterization. So this is utilizing a handheld cautery, also applied to the episcleral veins, and you get an increase in outflow resistance or decrease in outflow. And there are several permutations to this model in the literature. For example, the one I have pictured here is a subacute model that was described by Lane et al. In 2019. So in this case, you can achieve a very drastic IOP elevation if you choose. Ganglion cell loss can range anywhere from 20 to 40% over a period of 2 weeks to 8 months. The consideration here is it's a one-time only event. You can configure by the number of veins you choose to affect with the cautery. And so if you want less or more, that determines the magnitude of IOP elevation. There's a high incidence of secondary pathology, such as inflammation in the anterior segment that comes with this model, and a high level of skill is required. As with laser photocoagulation, there are species and strain variabilities. The third is circumlimbal suture, and this is similar to photocoagulation, except you're using ligation by suture of the episcleral veins, and this compromises the scleral outflow. And there are various modified methods for how to do the suture, what to use for the suture that result in various levels of IOP elevation. The one I have pictured here is Matthews et al., who described a modified model, version of the model for gradual IOP elevation. So similar IOP elevations to the other two we've discussed, similar RGC loss. There's a high skill level required, but it's less invasive than cauterization, and so secondary effects are lower. Okay, so injectables. This is everything you can inject into the eye. So the first one is hypertonic saline. And so this is the use, this is kind of my bridge model, right? Because it is the use of hypertonic saline injected into episcleral veins to induce sclerosis, right? So this is kind of similar to the models I just discussed. It impedes outflow in a similar way. IOP elevation is variable onset, and so that is something that ranges across the model. There's 50 to 150% elevation, depending. It's a rat model. There's a known circadian effect. So IOP pressures are higher in the dark. That's probably true for all models, but it's been documented in this one. There's low equipment costs, but there is a high incidence of secondary pathology, and repeat injections are necessary to maintain the IOP elevation. The second is microbeads. So now we're switching to injecting things into the anterior chamber. This is achieved by intracameral injection of microparticles, and this then impedes aqueous humor drainage through the canals. So various different types of materials have been used: latex, polystyrene, magnetic beads that then use a magnet to get greater seeding of the microbeads into the anterior chamber, and then also the combination of these type of particles with viscoelastic substance to, again, kind of seat them better in the canals. And so IOP elevation is much more moderate. It's about 48 hours to peak, 20 to 35% elevation. Ganglion cell loss is 20 to 35% at approximately 8 weeks, although it can be extended longer. The key here, one of the key considerations here is that repeat injection is necessary, usually at about the 3 to 4 week mark, and there are age and strain dependency, as we heard with the optic nerve crush. So different species and different strains will respond differently to the stress. The third is hyaluronic acid. Again, we're doing an intracameral injection, so it's going right into the anterior chamber. And in this case, the hyaluronic acid progressively increases anterior chamber depth as you do repeat injections of HA. So IOP elevations, 80% over a similar time period to the microbeads, about 2 days. RDC loss is approximately 65%, 8 weeks. And so considerations here is that, like the other two models we've talked about in the injectable categories, the benefit here is that there's kind of low equipment and startup cost. There's skill required with the intracameral injection, but again, we have re-injection necessary this time in the weekly time frame. The final is silicone oil. So this is intracameral injection of silicone oil that occludes the iris, and this is different in that the IOP elevation is in the posterior segment, okay? And this model is actually based in or recapitulates post-operative complications in humans. So the IOP elevation, the rate is unknown, really, but it's about a twofold increase in IOP elevation, but IOP measurements in this model requires dilation. And so once you dilate, you're kind of reversing the IOP phenotype. And so doing it early is a potential issue for this model, and so we don't really know what the rate is. And incidence of secondary pathology appears to be pretty low. So we've talked about, Elliott talked about the glucocorticoid-induced model, which I think is important, and I wanted to highlight here, because this is a model that bridges both, right? So you can use it for anterior segment, but you can also evaluate ganglion cell loss, which after 15 weeks is at about the 30% mark, which is similar to some of the other models that we've talked about. And there are modifications to this model that now address potential systemic effects and look at slow release or nanoparticle formulations that reduce the labor required to keep administering the drops. Okay, so the third is intermittent hypertension models. And so these are models that are looking not to create a permanent IOP elevation, but one to evaluate changes over with short duration. And so there are multiple options for for inducing this type of IOP elevation that includes cannulation or compression. And I'm going to move forward. Yeah. So important points on the inducible models is that there are they all have sources of variability, and they are all costly in some form, skill, time, or equipment. They all have pros and cons, but these are relative. And so the best model to use is the best model for the question you're asking. okay? And it should be driven by the hypothesis that you are testing. And also, if the best model does not exist for what you're looking to evaluate, consider developing a new model, right? All of the models we talked about were developed in response to evolution of knowledge in our field. And if you're experiencing challenges and not finding what you need in models, it's likely others are doing the same. Model development is challenging and rewarding. Tap into the ICER network here. Many of the model developers that I've talked about and others have talked about are present at this meeting and are happy to talk to you. Okay.
Thank you very much, Becky. Our last speaker is Colleen McDowell at the University of Wisconsin.
All right, thank you. So my talk is going to be focusing on considerations we should all have when using genetic and transgenic mouse models of glaucoma. And I have no disclosures. So I have used a variety of genetic and transgenic models throughout my career. Starting as a graduate student with Mike Anderson at the University of Iowa, we used the DBA2J mouse model of pigmentary glaucoma to not only study aspects of pigmentary glaucoma, but also as an inbred mouse strain to compare differences in mutations, for example, on the B6 genetic background versus the D2 genetic background. As a postdoc with the esteemed Abe Clark sitting right here, we did a lot of strain surveys. So using inbred mouse strains to detect strains that were more or less susceptible to different treatments. So this is just one example here where we were overexpressing TGF-beta2 in the trabecular meshwork to look at intraocular pressure phenotypes between different inbred mouse strains. And now in my own lab, I use a variety of transgenic models to look at ocular hypertension phenotypes and retina ganglion cell death phenotypes. And today I hope to convey to you how we can use things like spontaneous mutations like the DBA2J mouse, inbred mouse strains, as well as developing transgenic mice to study various aspects of glaucoma. So I always tell my students, our job is not to cure mouse glaucoma, it's to cure human glaucoma. So when you're using mouse models, you really need to consider what is the relevance to the human disease. So does the mouse model replicate key features of human glaucoma? Do you get an elevated intraocular pressure? Do you get retinal ganglion cell loss? Do you get optic nerve damage? And what is the progression of each of these phenotypes? Also considering disease mechanism. So is the genetic mutation on your transgenic mouse or your spontaneous mutation relevant to human patients? So is it mycilin, optineurin, TBK1, mutations that we see in the human population? Or is it part of a similar molecular pathway that might lead to new phenotypes or molecular pathways that might be relevant to the human disease? And also considering types of glaucoma. So what type of glaucoma do you want to model? Do you want to model primary open-angle glaucoma, normal tension, congenital? So these are all things to consider when you're not only picking a model, but also developing a model. What type of phenotypes do you want to see? Now, in my opinion, the most important consideration when using genetic and transgenic mouse models is the genetic background of the mouse. So I think Rob already showed us some interesting data from his work with optic nerve crush on the importance of genetic background. But we can't emphasize this enough that different mouse strains can vary in the susceptibility to glaucoma phenotypes. And one really great example of this is coming back to the DBA2J model. So as Rob described, I think a little bit in his talk, or I think there was some mention of it, that these mice develop elevated intraocular pressure that's progressive over time. This leads to progressive RGC damage and optic nerve damage. But if you take the mutations that cause these phenotypes in the DBA mice and put them on a BLAC6 genetic background, you lose that phenotype. So you have the same mutations on two different genetic backgrounds, one that causes severe glaucoma and one that does not. So you really need to consider genetic background when using these models and developing your models. And along those lines is ensuring that if you do backcross to another genetic background, that you do it for enough generations so you don't end up with a mixed genetic background, because that can really confound your results and cause variability between the mice. This variability between genetic backgrounds is due to modifier genes. So genetic background genes can influence the disease onset, severity, and response to interventions. But this is actually a really cool tool you can use, actually, to figure out new phenotypes or new pathways that might be modifying these phenotypes. So here are some examples of the ways you can use mouse genetics to identify cool new modifier genes that might be really relevant to disease pathogenesis. So you could do things like forward genetics with chemical mutagenesis. You can use more modern techniques like CRISPR-Cas9 to do a targeted approach to enable precise gene editing and screening. Probably my favorite way to identify modifier genes is actually just using the natural genetic variation in inbred mouse strains. So you have this existing genetic diversity among these individual mouse strains that can help you identify the modifier genes if done correctly. And there's a whole litany of computational and bioinformatics tools that you can use to help identify these genes in these studies. So this is a lot of words on this screen, but since you guys have access to these PowerPoints on the BrightFocus Foundation website, I just want to put this up here so you can come back to the talks and look at some more in depth on some of these tools that you can use to help your genetic and transgenic mouse model studies. So another important consideration is age and disease progression. So some models develop glaucoma really slowly and progressively over time, while others show a really rapid onset. So as Becky pointed out, the best model is the one that can address your hypothesis. So a rapid onset model might not be appropriate for the question that you are asking. In my opinion, second to genetic background, the next most important consideration is age matching. Age and sex matching your animals. So proper age and sex controls are crucial for interpreting your results, especially when you're looking at progressive changes in IOP and neurodegeneration. And another thing to consider is the window of intervention. So if you're designing an experiment for example, for neuroprotection, at what time point in your model is going to be most appropriate for you to apply that new therapeutic? Do you want to do it just prior to RGC loss? Or do you want to maybe do it even sooner before you start seeing early changes in the optic nerve head? So truly understanding the timeline of events that are occurring in your model is really essential. Another important consideration is off-target effects. So transgenes or knockouts may have systemic effects that you are not considering. There may also be compensatory mechanisms. So knockouts may affect alternative pathways that obscure true gene function. So there's been examples in the literature looking at a lack of neuroprotection, an example in JNK3 knockout mice. So JNK3 is a pathway that is known to mediate neuronal death signaling. So the hypothesis was, well, let's knock it out and maybe that will rescue neurodegeneration. Well, when investigators did that experiment, they found they didn't see any block of the neurodegeneration, and it's likely due to a compensatory pathway that made up for that loss of that really important signaling molecule. And then the next consideration would be promoter specificity. So is the gene expression restricted to ocular tissues, or is it broadly active? And this is important when you're looking at conditional knockouts. Are you truly knocking that gene out only in the trabecular meshwork, or is it getting knocked out somewhere else that might have some compensatory mechanism that you're not considering in the model? And then lastly, the consideration I would encourage everybody to have is in reproducibility. So colony management can have a big effect on these model systems. There's things called genetic drift. There's breeding errors that can change phenotypes over time. And if you're not properly monitoring or genotyping your animals, oh, I'm breeding these homozygous knockout mice, I'm just going to keep doing it forever and never go back and regenotype to make sure that there haven't been any breeding errors, that can really confound your results or make an investigator that you send these animals to, to spin their wheels and not realize why they're not finding the same phenotype. And the other thing is environmental factors, things like light exposure, cage density, diet, bedding. All of these things can have an effect on phenotypes, especially when we're looking at physiological things like intraocular pressure. So if you go in there wearing really smelly perfume and banging cages left and right, do you think the pressures are going to be high or are they going to be low? Right, so you really need to consider those things when looking at physiological points and different animal models. So this is a chart, again, a lot of words, but I just wanted to show some of the models that are available right now in the literature, and this is by no means an exhaustive list or a complete list of all the characterization that's been done on these mice, but I did want to provide at least a chart that you can come back to later to look at the variety of mouse models that are in the literature, as well to point out, again, that all models are not created equal. So many of these models have different IOP phenotypes, different timing of onset, level of IOP elevation, and concurrently different levels of RGC loss and optic nerve damage. So these are all important things to consider when choosing what might be the right model for you. And as Becky pointed out, there's advantages and there's limitations to all of these. I get asked a lot, what's the best mouse model of glaucoma? And I always say, well, there's not really a best one. There may not even be a really good one, but there's ones that are good to study different types of questions. So this is, not, like I said, not an exhaustive list, but I hope you can come back to it and look up some of these models and look into some of the advantages and limitations that you might want to consider in your own work. And then in the last minute or so, I did want to highlight a couple models that are most commonly used. So the DBA2J mouse model pigmentary glaucoma is probably the most commonly used one of all. And you have gradually elevated intraocular pressure due to mutations in two genes, GPNMB and TRP1. And they develop progressive damage to the optic nerve as well as the retina ganglion cell axons. And there's been many key discoveries that have been discovered using this mouse model glaucoma, and some of them are listed here. And again, I wanted to point out that there's a lot of information that was found here, and although this is a lot of words, many of these discoveries were actually done using different forms of mouse genetics. So knocking out various genes, like in this study where the Bax gene was knocked out and it was able to prevent RGC soma apoptosis, as well as things like lacking autophagy genes, overexpression of different genes. So there's a variety of ways that you can use existing models to test your own hypotheses. And then I also wanted to touch on the transgenic myocilin glaucoma model. So myocilin, as many of you probably know, is the first primary and open angle gene that was discovered in humans. And this particular model overexpresses one particular mutation, the Y437H mutation. And it was found that these animals have elevated intraocular pressure, progressive retina ganglion cell loss, and optic nerve damage, as well as some functional defects. But there's also been some important lessons that have been learned from this model. And one of them is that I emphasized early on, that genetic background is really important. Because some studies where this mutation was placed on the BLAC6 genetic background showed little to no phenotypes, while when the mutation was on various mixed genetic backgrounds, they had a more robust phenotype. And just in the last month or two, there's been a recent report that suggests that this model does not have phenotypes on 9 different inbred genetic backgrounds. And that data really indicates that there's an undefined complexity and that the previously reported glaucoma phenotypes may not be robust across all institutions and environments. So really the big lesson learned from this is always validate your mouse models in your lab and make sure you know what you're working with before you move forward. So I have given similar talks at the prior fast tracks where I went into great detail on a lot of the other mouse models that I had in that big chart. So because I don't have enough time today, I would just like you to refer you to the BrightFocus Foundation's YouTube channel where you can go and listen to some of the other fast track talks that we go into more detail. And then I just had a few take-home points, but I think those were all well discussed in the time I had here, so I will end with that. Thank you.
I would like to thank the speakers very much for their insightful comments and also for staying on time. So we have about 12 minutes for questions. So
Before we go to questions, is Jillian in the audience? You haven't uploaded your presentation yet, and you're next. Okay. All right, we might have to shift the order.
oh yeah,
So, Becky, I'm not sufficiently
would you? Yeah, it'd be easier for us to hear. Thank you. Lots of questions.
the circumlimbal suture model, whether or not it was written there, but one of the things I find really attractive about that is the controlled reversibility.
Yes, yes, exactly, exactly. Mm-hmm.
Mike.
On? Is that on?
Oh, there it is.
Yeah. So your talks were fantastic, and I think you bring up, well, you probably didn't even mention it, but I think you bring up a really interesting point. And Colleen, it was great to see all the different model systems and the genetics and stuff like that, because there's not a model out there that really represents everything that's going on. And I think this gets back to Yvonne's talk, is that there's such a diversity in the human population that the genetics play a huge role and the strain differences play a huge role. And so there's probably never going to be a model that represents everything that's going on. We know that there are patients that have high IOP but never get glaucoma. All right, somehow they have a genetic predisposition to, guarding themselves from that. But I guess the interesting question, and I'm trying to figure out how we get to some of the, the really key modifiers. And I know Janie's done a lot of great work on the genetics and everything, in humans. But I was really intrigued, Colleen, when you said that when you put the D2 mutations on a black 6 background, that they did not show the elevation in IOP and they didn't show glaucoma. Was that taken to the D2 model and back crossing to the black 6? Or if you put the mutations in the black 6, can you back cross to the D2 and then get glaucoma? Because I think that gets a lot with positioning of the genes and potential, key modifiers that could be involved in the disease.
Well, since Mike's holding the microphone, and that was Mike Anderson's work when he was at Jackson Labs, I will allow him to answer that question.
Hi, Mike.
Hey, Mike. Mike in the morning. So that was half of my postdoc with Simon John at Jax, was making N10 congenics for those two and then crossing them together. So they get the full-blown iris disease. You can't really tell apart from DBA2J. They just don't get high IOP. And then Cy spent half of his postdoc trying to map that modifier, and that's not been published. So it's a complicated multigenic story. Actually, I had a question as well. So I really enjoyed those talks. It would have been a lot simpler if the mouse genetics community had only built one mouse, but they didn't. So we've got all these different strains to choose from. And two of the most common are C57 black 6J and C57 black 6N. N because that's what the comp community has made all the knockouts on. So lots of other fields use those. And if you're just looking for a knockout at your local university, there's a pretty good chance that it's going to be on in the 6N background. And 6Js are just what most folks are more familiar with. But they both have a caveat that the 6Ns have that well-known caveat that they carry RDH, so they get a photoreceptor degeneration. Black 6 also have an odd mutation in this NNT gene, which influences mitochondria, and it's related to nicotinamide. the diabetes field doesn't like to use 6Js for that particular reason. I was wondering if anyone had any actual hands-on experience comparing those two strains and whether the models that you spoke about, either one of those mutations had an exact influence. You could suppose that they're both less than ideal, but maybe tolerable. But I was wondering if anyone had a better answer to that.
I do not. So, I mean, I primarily just worked with the 6Js, and if anybody had any experience, I guess it'd be you, Mike, right?
So Jennifer, who's in the room, will hopefully have some experience because a mouse that we just received is in the N, and we're in the process of backcrossing it to J. So, and we're going to do some experiments also on the mixed cross. So we don't have any, other than getting rid of the RD8, which was our initial thing, we need to get rid of the RD8. We are generating sort of a mixture of those backgrounds, and hopefully we'll have both at some point.
So they also have an alternate immune system as well, about 6 mice. So I have a quick question. So there's a lot of variation in the animal models, and relatively new to this field. Where do people go wrong when they're working on their projects and writing grant applications? So let's say a reviewer might just decide this is an inappropriate model for this study. What are the things people should really look at and avoid in that context?
So have you taken the recent conflict of interest, how to do reviews for the new scoring system in the NIH? Because this is one of the issues that they play out in the course that they give you. So one of the ways you go wrong is you have a reviewer that hates the model that you're using. And it's that kind of basic bias that can really trip you up. So you have to be very careful when you're saying you're going to use the model, you can specify how it has limitations, but how it's important for what you need to study. And there's a lot of personal bias that comes into glaucoma research specifically because of the models that you're using. So it's a matter of wordsmithing, I think, more than anything.
And I would say, Colleen said it beautifully, and it's outlined in the slides on mine also some examples of being aware of what it is you're looking to target, right? And make sure that the model that you're using is appropriate for that with respect to onset, duration of time. If you're studying inflammatory signaling, you don't want to use a model with high secondary inflammation, right? And so hopefully a reviewer will be operating not from bias, but it happens. But if you're clear on why you're choosing that model and can defend it, you should be good.
Question way in the back. Barry, thank you for being patient.
Thank you all for a series of fantastic talks. My question is almost very naive and basic at this point. I've always had trouble convincing basic researchers about why I use mice where the proportion of conventional outflow through the Schlem's and the trabecular meshwork to that of the uveoscleral is so much skewed in the opposite direction than compared to humans. Can you comment on that? Is that a very naive standpoint? And how do I convince somebody that mice can indeed, the compensation that they have with the uveoscleral is in fact comparable to what we see in humans?
So I can't comment directly other than that as I was preparing this, I found a very recent paper from Ross here. Ross isn't here right now, right? But he might be here, where they're making very discrete measurements of the volumes of the chambers and actually come to the conclusion that uveoscleral is actually pretty low in mice. So you might look at that or talk to Ross.
There has been conflicting reports out, but I think the consensus now is that in fact it's very similar to human. That the major outflow is through the conventional pathway, trabecular meshwork, Schlem's canal, and uveoscleral is 20 to 30%.
Yes. Hi, thank you guys for the great talk. I had a question about, there's been mentioned a couple times that you want to limit the amount of inflammation that your model produces, and this is a super naive question, but I'm wondering why that's a goal, if potentially the inflammation could help with some of the TM changes and then IOP elevations in these models.
You want the neuro answer or the
Right.
anterior segment answer? There are two different answers.
Well, I'm assuming your question's coming from someone using like Ad5 versus lentiviruses, or? Oh, well, I mean, in the context of like the front of the eye, I would not recommend using microbeads to study trabecular meshwork outflow, because you're artificially clogging the drainage structures. So that's not really going to be a true indication of what happens to those tissues in glaucoma. But I'll turn it to Becky for the rest of it.
I don't know why. So the key, I think, to lowering the IOP is not, or lowering the inflammation is not so much the inflammation related to the disease process, it's the inflammation relating to the induction method. So if you are wanting to look at or facilitate even inflammation that is part of the disease process, you're looking at it here, right? But that's going to be relative to whatever your background is. And if your background is super inflammatory because it's a response to your induction method, then that makes it more difficult for you to actually evaluate what's happening in the landscape of inflammation. So it's more about, kind of clearing the background picture. But you are absolutely right. You don't necessarily want to impede what you're trying to study.
So Yvonne?
Thank you all for some great talks and instigating or inspiring us to try to develop new models. I have a question for Rob and also the panel. You mentioned the contralateral eye not being an appropriate control, which I agree with, and there's definitely issues with that. But how do we balance that as investigators, trying to reduce the number of animals we use? Do you think that comment also makes a difference whether or not the particular outcome measure you're trying to examine may or may not bias you into finding an effect? For example, like if you are looking at ganglion cell loss as an outcome measure and your control is also showing some ganglion cell loss, then perhaps would you say that depending again on what you're trying to identify, might be, you're not biasing yourself for finding an effect, you're potentially minimizing or decreasing that, if that makes sense.
No?
So what I'm saying is that if the contralateral eye is showing some ganglion cell loss, for example, and you are looking, let's say, at a neuroprotectant for finding a benefit, you're treating the contralateral eye as the control, and you're treating the glaucomatous eye, then if you find an effect, if you have already, if you do find an effect, I don't think you're necessarily biasing yourself in the wrong direction.
True. You might even be erring on the side of a more robust result, right? Yeah, I agree with that. But when we talk to our graduate students about what a proper control is, and we want to tell the community, like, this is what normal looks like, and this is what. And I don't do a lot of neuroprotection. I do more of what are the nuts and bolts of the pathology kind of stuff. So in my mind, when I want to say what normal is, I can't show them a contralateral eye because the data I'll show you later, it's not normal.
Right.
And in fact, you and I need to talk because you're on my list to ask my 4 questions to.
Okay.
And I agree with you, and we've learned many great things using the contralateral eye as our control. So I think you just need to be careful.
I know. Yeah, you have to be very careful, and I just and also am thinking about in the context of animal numbers, and that issue also becomes more and more challenging.
Right.
So I do actually want to make a comment on that too, because I think it's an important point, right? Is it then actually part of the glaucoma model, right? In a human who has a normal eye, right? Or it's not, right, necessarily normal, but that is actually then that mouse becomes more representative of a unilateral glaucoma patient. Right, because there is, I think, the caution flag of don't throw away the contralateral eye, right? It's relevant to potentially glaucoma pathology and what we understand about the disease and worth studying as that, right? Yeah.
I would like to thank the audience very much for your great, terrific questions. I want to respect our break, so I would suggest if you have any additional questions, you come up and ask the individual presenters. So thank you very much. We'll be back at 10:45.